Our next speaker is Sergio Ruiz. He has several years of experience discovering, analyzing, vetting, and propelling longevity biotechnologies that can move the needle in the mission to increase the healthy human lifespan. He's also an entrepreneur, investor, who prides himself in helping with the incubation and formation of young academic teams and developing them into industry leaders. Aside from being the co-founder and managing director of the Methuselah Fund, he currently holds several senior advisory roles for longevity biotechs, is on the board of directors for Leucadia Therapeutics, and is a co-founder and chief operating officer for a Stanford-born company called Turn Biotechnologies, focusing on epigenetic resetting of the age in a cell.
Thank you, Daria. Good to see so many familiar faces. Thank you, Christine, for setting up this amazing panel and presentation. So over the last several years, we've been focusing on epigenetic reprogramming with the goal of getting products out to people, not just understanding the science, but how do we make it translatable so that we can actually help human beings that are currently suffering. And we've been very stealthily and very dedicated to rigor in science. A lot of you have interest in understanding where we're at, especially since the field has been evolving with incredible players and incredible funding. And it's my pleasure to try to give you the most up-to-date data today. Now, bear in mind, I'm just a business guy who stumbled upon an understanding of longevity, and someone who believes that longevity is ultimately the spearhead of innovation in the next generation of healthcare and medicines. So I'm very excited to present some of the data. Now, I see there's a tremendous amount of understanding on epigenetic reprogramming. There's a big spectrum of knowledge, so I'm going to try to be as generic and as deep as possible. So my goal is, if I can add value by giving you something new, something interesting, something fun, then I've done a good job today. I'm happy to talk to anyone afterwards and explain how it is that we are focused on getting products out to people. So, let's see if I can move this along. We started with a pretty simple observation. People are not only living longer, but also getting sicker for longer. Now, I'm an economist by training, and I understand that that's going to bankrupt the modern world as we know it. Something has to be done. And what we realized was that current therapies were not just incremental, they lacked the exponential innovation that you could see in other industries, but they were also masking conditions. They were a way to not solve, but mask problems, pain, and suffering. So they did a poor job at a cost that was not sustainable. So I always like this slide because it just shows the impact that solving aging could have, or actually solving aging-related diseases. What we see is that chronic disease is an actual malady of the modern world. We see, so we started looking at the cell and trying to understand what is it that breaks down and how can we fix it. Now, a lot of you understand the hallmarks of aging, but of course, the biggest things that we realized was that, basic functions like protein production, efficient cell division were just completely becoming lossy, or becoming, I guess, the performance was going down and degrading with time. So you can actually visually see what in skin, what healthy cells and what defective cells look like. It's a very visual understanding of the degradation that we all go through. So we start looking at how to crack the code on cellular degradation. We call it health span engineering, and within this context, epigenetic engineering. And this is done with technology that is well known, epigenetic reprogramming technology has been widely studied. We decided to use messenger RNA because it's something that is highly tunable, it's something that is highly understood. And even before the pandemic, we realized that this was going to usher in the next wave of therapies that could be not just safe, but could go to market in a pretty great time frame. Of course, the pandemic allowed the FDA to understand mRNA and delivery of mRNA in a way that has never before seen at a speed that was just incredible. But there's two main observations that we noticed. We noticed that cells, young cells, when you rejuvenate them, they don't just perform better, but they make everyone else better. I have a term that I'm still looking for a better terminology, so if you think of one, help me out afterwards, tell me, but I call it gentrifying the environment. So what happens is you rejuvenate the cells, and even if you don't rejuvenate 100% of the environment, if you go even down to 10% of it, that 10% takes over and rejuvenates the entire tissue. Of course, an entire tissue means rejuvenated means a rejuvenated organ, a rejuvenated set of organs, a rejuvenated system, and that's the ultimate goal, to be a little bit more systemic as we go through the motions and take products out to market. We also see that the cells that are rejuvenated get a restoration of their youthful performance. And despite the fact that we have data in 40 different types of cells, we're going to focus on two of our leading programs. Again, our goal is to get products out to people. And that's one of the programs is in immunology. So we're going to talk a little bit about how T cells are enhanced by our therapies, and also how skin cells are going to be addressed. And we actually had an announcement earlier today with the goal of telling the world that this technology is now far along, that we can talk to dermatologists and they're getting excited about this coming into the clinic. But before we get to the data, I want to explain how do we get to the clinic? How do we ultimately deconstruct making a product from a new technology that could be seen as unsafe and then put it into a package that can actually do good? So we use three pillars, of course, the modern tools, machine learning, big data, understanding via AI and such. So we have a bioinformatics component to what we have. So we have 8 years worth of training that we've been doing in different kind of cell environments, different kind of cells, different kind of delivery mechanisms. And all that together paints a picture that it's a little bit more sophisticated and it's able to educate the regulatory bodies in a much more efficient way. And we're very excited about it. We also focus on cellular tuning. Very early on, we decided not to use viral vectors because we didn't want to worry about DNA integration and different kinds of tumorigenesis, oncogenesis. So we focused on mRNA, and then we decided to innovate mRNA. We actually have these little things we call TuRNAs, turn, and we fine-tune them to the environment, to the cells that we're targeting, and we have different ways of approaching via polycistronic, monocistronic applications, depending on how much we want to control the half-lives. Now, we can control the half-lives of the mRNA, so what that essentially means is that we know how long the reprogramming will last in a certain cell, and that's how we know that it's not going to turn into a dedifferentiated cell or into a cancer cell or some kind of zombie cell that we don't want in our bodies. And then the next stage is what I call 50% of the equation. You can't really put naked mRNA into your body and expect it to reprogram you into a youthful version of yourself. You also can't really use off-the-shelf even LMP-approved, FDA-approved solutions for a lot of the indications. And we know this because we've tried. And transfection is an important thing for delivery, but also safety. And what we've seen is that there are some mechanisms of delivery that are lipid-mediated that are toxic. And while they might be okay for a vaccine when it's about to save your life because, the world is undergoing a pandemic, they might not be okay for trying to make your cells young in different applications. So I'm going to bypass this one a little bit faster because I talked about it already, but our cocktail, we call it ERA, Epigenetic Reprogramming of Aging. It's really just 6 genes that have been tuned and optimized. At the core is the Yamanaka factors, but on top of that, there's a sort of tunability that, again, it's addressing different cell types, but also different environments. And of course, we have an interesting ability to control not just the half-life, but the immunogenicity. We can do different kinds of fun experiments with activating certain genes versus others and timing what gets activated first and last and different things like that. Of course, I can't go into detail, but that's something that we've developed in the last 4.5 years. And then the precision delivery, again, we had to go and create our own novel lipids. So when 2020 hit, we realized that we were competing for material with the biggest companies in the world that were making vaccines. So not only did we bring the mRNA production in-house, which allowed us to create our own better purity, better material just altogether. We were able to bypass the supply chain problems by creating old plasmids and such, but also we invested heavily in several chemistry departments, analytical chemistry, synthetic chemistry, to understand how to put all this together into a product. So we started looking into different applications, indication, and the question was, if you can reprogram any cell in your body, which one do you want to reprogram first? So we decided to do 2 main ones. Dermatology, and I think a lot of you have told me before, it's a great indication. Every time you look at the mirror, you could be a walking advertisement for rejuvenation. You understand that now your skin is looking younger, but maybe you ate something that you used to digest very well, and now you're not digesting it the same way that you used to in your 20s, so then you start thinking, what else can I rejuvenate inside me? So it's a gateway into the zeitgeist, the understanding from a societal point of view of the need for these therapies. Also, it was a fun example of our in vivo approach. There's different modes of administration that were already understood, and delivery in itself is actually quite accessible because it's the largest organ in your body. It's very, very accessible via different kinds of methods, but also is very well understood by the FDA. And it gives us a very nice regulatory path. And then immunology was an exercise in trying to figure out how do we deliver ex vivo into FDA-approved therapies that are now stratospherically expensive. For example, CAR T therapies. I think the average is between $2.5 million, and every 2 months you hear of $3 million and then a $4 million therapy that's coming out, all with surrounding CAR T therapies. And we said, well, what if we deployed our technology ERA here? Could we make it cheaper? Well, you'll find the answer very soon with our projections. And of course, being a company, we need to make sure that we have a way to stay in business and address it. So, ERA as it pertains to immunotherapy. I'm going to do my best to explain the science, not being a scientist. But really, the problem with immunotherapies are not just the cost, but also something that is a rate limiter called cell exhaustion. So despite how old you are, if you have cancer, if you've undergone chemotherapy, radiation, by the time you get this $2.5 million therapy, your poor soldiers that I like to call, the T cells, have been fighting against this cancer, and they're showing an aging phenotype. So again, the thought process, if we tackle aging, could we tackle things that are not necessarily attached to chronological aging? And that was what we found out, was that we can actually enhance proliferation. So your T cell super soldiers are, your army gets multiplied by 5 times. That's what we're seeing. And also their ability to kill the cytokine storm that it creates to kill the tumor specifically also goes up by an average of 4 times. So compounding all that, you have a therapy that conservatively could reduce the cost by 75%. It's scalable, and I can explain that in a little bit, how that works. So what did we decide to do? We decided to test this. We have the standard model, very well understood by the FDA. You harvest the T cells, you add the CAR T therapy, put it back into the patient. So we decided to do an experiment, and this is one of many, where we decided to do engagements of tumor cells into treated cells and non-treated cells, non-ERA treated cells. So you'll see the standard cells go through several engagements, 6, and so do our ERA-treated cells. What we start seeing is that at the 3rd battle, right, the non-treated cells get exhausted. And then beyond that, all the ERA-treated cells are still fighting the war and eventually win the war. And that was very exciting to us because we saw everything just became more efficient. That entire understanding on how to fight that cancer became a lot more clear to the therapy. So T cell proliferation went up, tumor cell killing started going up after, it started being differentiated as a better solution after the 3rd engagement. So how do we figure this out? So there's certain exhaustion markers that you can measure, and these are the ones that we measured. And then also there's certain proliferation markers that we saw, and they were all being expressed. They were just the soldiers that could not get tired. And we started comparing it with the industry. What are others that are trying to fight cell exhaustion doing? And some of them are trying to use cellular reprogramming, and we saw the differentiation from some of their data that they made public. But this is a key factor because the T cells maintain their identity. This is what everybody asks. What if trying to solve cancer, you create more cancer? Well, we didn't see that. We maintain the identity, and we know that because we measured the markers for that cell identity. So how does that translate to us in a daily basis, right? Again, our focus is to get products that help people out to them. Well, we realize by using some actual examples like Yescarta and Chimera, conservatively, we could turn a cost per dose down significantly. And this is for autologous therapies. We're very excited about this because very conservatively, we can deploy this into something that's already FDA approved, making it so that all these therapies can help people better. Now, we're not necessarily going to go out and become an immunology company. We're at the core an innovation company using longevity as a way to get there. But we see a world in which all the CAR T therapies and all the new NKT therapies and similar therapies, immunotherapies, are going to need this kind of rejuvenation to avoid the cell exhaustion. So, and then one more thing that I wanted to point out, there's a whole new industry that could potentially open up, and that's the patient banking of future doses. Because if you go through fighting cancer, you may be able to bank them just in case it comes back years down the line. Currently, there's a lot of waste. If you don't need it anymore, all that goes to waste. But with this model, we could potentially just bank those really good soldiers, super soldiers, for the future. How about dermatology? So in dermatology, we decided to just attack something that has been understood for a while. It makes billions of dollars, but the best in the market is actually not causing rejuvenation. It's really just an acid that creates some kind of damage that creates a rejuvenative effect, but it's not really true environmental rejuvenation. So how do we do this? How do we go about proving this? Well, remember, our story is not just a mRNA story. It's a delivery story. So we start with delivery in vitro. Can we actually take our novel lipids, our proprietary delivery, and deliver, right? Now, I would say that that we can. I mean, you have the pictures there. But then we're start looking at different rejuvenative markers, and we are going to focus on two: metalloproteinase and SAW2, which is a oxidative stress marker. And what we saw was actually a decrease in dermal fibroblast markers of that break down the skin, ultimately. And now what we see also is on the opposite end, this biomarker H3K9ME3, which is a rejuvenation marker, went up significantly. What we ultimately saw was that on an environmental level, the benefits were compounded. And anything that was overexpressed was downregulated. Everything that was underexpressed was upregulated. And it was very exciting to see the cell proliferation as well. We pretty much had better bricks and better mortar, right? The elastins, the collagen were just rejuvenating the dermis, the epidermis. And then we got some great in vitro conclusions. We went into ex vivo and decided to do different delivery experiments there. And we were successful at targeting exactly the cells that we wanted. And fast forward, we saw that our TLP formulation, TLP is what we call our delivery lipid-mediated formulas, are actually doing a much better job than Renova, which is the industry standard for rejuvenation. And we got our ex vivo conclusions and ultimately decided to go in vivo. This is where we stopped publicly, and this is where our focus is from an education perspective and a pre-IND package perspective to educate the FDA on our therapies. We have a clinical pathway. We want to be in humans as soon as possible, so we're predicting early 2024 after we finish all the submissions and then GMP manufacturing processes. Just a little snapshot from the company. And just to finalize, I asked chat, the OpenAI chat bot, one of the best open AIs out there, that to write a poem about epigenetic reprogramming. It was really interesting. I'm going to read the last part here. It says, from healing From the healing of a wound to the birth of a new organ, epigenetic reprogramming holds the key to unlocking the mysteries of life. So let us dance and let our genes express in ways that are true and bold, for the future of a species lies in the secrets that we hold. So if an AI understands the importance of rejuvenation via epigenetic reprogramming, how could we not? So thank you for your time.
I think in the interest of time, we'll break now, and you guys can find Sergio over coffee.